In this class we are going to talk about electrophoresis and SDS PAGE. They are not really a downstream process, but they are techniques which are used for purifying proteins and identifying their sequence as well as trying to understand their structure. So, they are very useful techniques used in analytical processes. So, what is electrophoresis? It is used for separating and purifying proteins and nucleic acid based on their size, charge or conformation. It is used mostly, as I said, in lab, biochemistry lab or molecular biology lab. So, what you do is you try to move the proteins, the charged molecule in a solution by applying an electric field across the mixture. So, based on the charge that is present on the protein, based on the electric field you are applying and based on the time, proteins separate out either in a one dimensional or in a two dimensional matter. So, we will look at it in more detail. So, what is the principle? It is based on the charge, according to charge. When charged molecules are placed in an electric field, they will migrate either the anode or to the cathode depending upon the charge that is present on the protein. According to the size, so smaller molecules will run faster than the larger one. So, smaller molecules will try to go faster, whereas the larger molecules based on molecular weight will try to move slower. So, the velocity of the charged particle in an electric field is directly proportional to the voltage, the charge on the protein. So, higher the voltage, higher will be the charge on the protein. The same protein can have different charges, then inversely proportional to the distance between the electrodes, how far the electrodes are placed and also the size of the protein. So, larger the protein, velocity will be lower, smaller the protein, velocity will be faster. So, they are given by this relation, velocity v on the left hand side, d is the diffusion coefficient, okay. X is the charge on the protein, phi is a constant, this is given by 96,500 current per mole, delta v is the voltage difference and this delta is the distance. So, as you can see, the velocity is directly proportional to diffusion coefficient, directly proportional to the charge and directly proportional to the voltage you are applying, but it is inversely proportional to the distance between them. And the diffusion coefficient is a function of the size of the protein, okay. It is function of size of the protein, viscosity of medium and so on. So, if I have two proteins with the different charges, then they will move at different velocities. So, after a certain time they will be attaining different distances from the starting point, and that is what it is, that is the principle by which they are separated. So, the velocity of the protein 1 will be v1, so v is given by this. So, that v1 is calculated based on the charge on the protein as well as the diffusion coefficient of protein 1. So, velocity of protein 2 will be v2, again v2 will depend upon the diffusion coefficient, that is a inversely function of the size and also the charge. So, the difference in velocity will be delta v, v1 minus v2. So, the distance will be, distance travelled in certain time will be delta v multiplied by time. So, if I give longer and longer time, I will achieve longer and longer separation, okay. And so, the separation depends upon the diffusion coefficient, which is inversely proportional to the size and directly proportional to the charge the protein has acquired, okay. This is the principle by which this type of separation takes place. So, what are the instruments and reagents you need? You need a buffer, you need a power supply of course, supporting media, detection and quantification, okay. These are the various reagents and instruments we require. So, the buffer, this carries the applied current, this establishes the pH, determines the electric charge on the solute. So, high ionic strength of buffer produce sharper band, low, but it produces very high heat. So, temperature could be very high. So, commonly used buffer are barbital buffer, tris EDTA for protein, tris acetate EDTA and tris borate EDTA 50 mole per liter for a pH of 7.5 to 7.8. So, these type of buffers are the commonly used. So, support material used in electrophoresis, it could be paper, it could be starch, it could be agarose, polyacrylamide, cellulose acetate and so on. Some of them are hydrophilic, some of them are hydrophobic. So, we can use different material depending upon our requirement. So, agarose and polyacrylamide gels are cross linked sponge like structure. So, the support media should be electrically neutral, because you do not want the voltage or which you are applying to get drained. presence of charge group may cause migration retardation. Okay. The flow of water toward one or other electrode is called electroendoosmosis. This decreases the resolution of the separation. Okay. So, if there are any charge group present in your gel, that is going to retard the movement of the protein, because the protein are moving because of the voltage that is applied at a distance on the two plates, and also if the water flows from one or the other electrode, then it is called electroendoosmosis. So, it decreases the resolution of the separation also. So, these two are problematic. In addition, I also mentioned that heat generated is also a problem, because if you apply very high voltage, you will get very sharp peaks, but then heat is also liberated. What is agarose? It is a chain of sugar molecule. It is extracted from seaweeds. It is very good for separating large protein, polynucleotides, 50 to 30,000 base pair. So, the pore size is determined by adjusting the concentration of agarose in a gel. Normally, it is in the range of 0.4 to 4%. Polyacrylamide, it is a cross-linked polymer. Okay. It is a synthetic polymer. The length of the polymer chain is dictated by the concentration of acrylamide. Generally, it is about 3.5 to 20%. These gels are difficult to prepare than agarose gels. Because oxygen inhibits the polymerization process, they must be poured between the glass plates. Okay. So, we can go to agarose based gels, or we can go to polyacrylamide. Polyacrylamide gels have a small range of separation, but it has got very high resolution. In the case of DNA, polyacrylamide is used for separating fragments of less than about 500 base pairs. In contrast to agarose, polyacrylamide gels are used for separating and characterizing mixture of proteins. So, if you want to characterize large number of proteins, then this is good. So, we have one dimensional electrophoresis, and we also have two dimensional. I will briefly touch upon that. So, the one dimension electrophoresis consists of SDS PAGE, isoelectric focusing, and native PAGE. These are the three techniques. Okay. SDS PAGE, isoelectric focusing, and native PAGE. So, SDS PAGE for separating proteins by mass. Proteins are denatured in detergent. So, why do you call it SDS? Sodium dodecyl sulfate. This is surfactant, sodium dodecyl sulfate. So, what it does? It denatures the protein. So, the protein sort of binds, or it forms agglomerate. So, that is what happens actually. SDS denatures proteins by binding to the hydrophobic core of the protein and gives each protein a negative charge proportional to its mass. Okay. SDS binds to proteins in a ratio of 1.4 grams of SDS per 1 gram of protein. That is how SDS works actually. So, basically you are denaturing the protein. That means protein will lose its 3-dimensional structure, and the SDS attaches to the hydrophobic core of the protein. So, by doing that, there is a negative charge given to the protein based on its mass. So, the separations will be based on the charge, which is in turn based on its mass. Okay. The protein may be further treated with reducing agents such as dithiooerythritol to break any reformed disulfide bond. Basically, you want to break the protein. So, when you do SDS PAGE, the protein loses its conformation, so loses its activity. Alkylated with the iodoacetamide to prevent further reformation. So, the end result will be a denatured protein, it becomes linear. So, all the proteins will have similar structures, and so they are separated based on their molecular weight. So, in SDS PAGE, you are separating proteins only on the molecular weight. So, you are not looking at the 3-dimensional structure, conformation, and so on actually. So, unlike conventional gel electrophoresis, where protein would need to be broken into linear portions, in SDS PAGE we can look at the entire protein in the linear form. That is the advantages. Okay. So, typically this is how it will look like. Okay. You have the molecular weight markers here. So, we can buy standard molecular weight markers, and each marker will represent a certain molecular weight. Okay. Then we have the product mixture, we will load it here. So, generally you load about 10 microliters of about 2 mg per ml denatured protein per sample, and this, when you apply the voltage, protein after certain time proteins will start traveling upwards, and each band will represent a molecular weight. From this molecular weight marker, we can read out what type of molecular weight proteins I have in this mixture. But there are many issues. Sometimes it is called overloading, sometimes it is called underloading. So, for example, this is overload, precipitation and aggregation of proteins. So, it is producing streaks and smears. This is an underloading, you have not loaded enough protein here, so we see very light bands and only the most abundant polypeptide is detected. Okay. The less ones are not detected because you have underloaded your protein. What are the stains for detecting protein in polyacrylamide gels? We can add 0.1% Kumesi blue dye in 50% methanol, 10% glacial acetic acid. Acidified methanol will precipitate the proteins actually. A doublet, if you have an SDS page, it indicates presence of 2 polypeptides that share the same amino acid sequence. Okay. So, a doublet. One may have extra residues and or they may differ in a few key positions only. So, they are part of a family. So, if you see doublets. Polypeptides from cytoplasmic proteins or that are membrane associated but extrinsic to a membrane tend to form well resolved narrow bands with sharp edges. So, ideally we should see narrow band with sharp edges, then we can say they are completely cytoplasmic proteins. Polypeptides that form intrinsic proteins bear sequence of hydrophobic side chains that may or may not denature completely. So, they will form bands that are broader and the edges will be less distinct. Bands that consistently show up following electrophoresis of a particular sample, that means the sample is characteristic of that particular polypeptide. So, that polypeptide, that sample is characteristic, so it keeps coming and again when we run electrophoresis. Bands that occasionally appear and or rather faint, it means you have some important polypeptides or something else which we do not know, and large proteins degrade, okay, when they degrade into 2 smaller products and each resolve into a band. Okay. Disappearance of a heavy band is correlated with appearance of 1 or more smaller bands, that means degraded or degradation of that large band which has led into something into smaller fragments. Bands containing 2 or more polypeptides, that means presence of polypeptides that are normally form non-covalent association with each other, okay, or polypeptide is present in high relative concentration. Okay. So, these are various techniques by which you could conclude by looking at those bands that are appear in your SDS. For example, if you have a hemoglobin native form, it consists of 4 monomers each with 15 kilodalton, they are all associated with the heme group. So, when you run that, you will see 4 bands corresponding to monomer, dimer, trimer or tetramer of the hemoglobin. So, we will have 4 different bands each having different molecular weight and each one will be characteristic of the monomers present in the tetramer. So, commonly used protein stains, okay, if you want to detect protein at the 1 to 2 microgram or in the nanogram range, okay, so we can use different types of stains. Okay, we will not go too much into that, but as you can see for detection in the nanogram level or in the microgram level, we can have different types of strains are there actually. Okay. So, if you want to stain nucleic acid, then we need to use a fluorescent dye like ethidium bromide. So, large number of protein stains are available. Then comes isoelectric focusing. We all know what is isoelectric point, right? The pH at which the protein completely loses all its charge. Okay. So, it passes through a gel containing a pH gradient. So, the charge on the protein depends on the amino acid chains. These charges change with pH. The protein will keep moving through the pH gradient because of the electric current and because of the charge, but as soon as the charge is neutralized, when it reaches the pH, isoelectric pH, it will not move, okay, it will stop there, and that is how you achieve separation. So, if you have proteins with different isoelectric pH, they will move in a pH gradient and then they will stop as soon as they reach a neutral pH or neutral charge condition. Immobilized pH gradients are used for isoelectric focusing, okay, because the fixed pH gradients remain stable at very high voltages. Buffering compounds are used to create the pH gradients. Okay. Immobilized pH gradients are cast strips of gel with plastic backing sheets. They offer high resolution reproducibility and allow high protein loads. Okay. Then comes native page. Okay, the advantage of native page is we can get the protein in its active state, unlike the SDS page we completely destroy the activity of the protein and make it linear. In the native page the protein still retains its activity, okay, because you are separating based at native states, you are according to the differences in their charge density. Native state of a protein is properly folded state, so it is not denatured, okay. But if you are adding denaturants, then the protein or buffer proteins or gels, then it lose its native states, okay. So the native protein we can take out the protein and then look at activity using some biochemical assays, whereas in SDS page the protein loses its activity completely. Native page is used for preparation of purified and active proteins, okay. So in the lab if I have many proteins, I want to recover a particular protein with the activity. So sometimes I will run a SDS page, look at the molecular weight, then I will run with the native condition. So it can be carried out at neutral pH to prevent denaturation, because acids and bases can denature a protein, okay. So we can study individual conformations, aggregation, self associations and so on, and if you need to carry out better in cold condition, the apparatus should be in the cold condition, so that the protein does not lose its activity. Okay, now we talked about one dimensional, now you have two dimensional electrophoresis, okay. The proteins are separated right angle to each other in two dimensions. The first dimension could be isoelectric focusing, that means you may separate the proteins based on their net charge. The second dimension could be based on their mass. So initially you separate based on charge, then you run a SDS page, so that the proteins get separated based on their mass. So instead of getting a linear protein movement, we will get a protein movement in two dimensional, okay. So and then we can pick out the protein of our interest and go use a MALDI or a mass spec and then find out what is the molecular weight of that particular protein. That is the advantage of two dimensional electrophoresis, okay. So you run the isoelectric focusing, that is the first dimension where the protein is separated based on charge. In the second dimension the protein is based on the molecular weight in the SDS page, and then you can look at the molecular weight of the protein. So proteins are not resolved as bands like in one dimensional SDS page and so on, but it is resolved as spots. So you have pH in one direction and you have molecular weight in another direction, okay. As you can see, they get separated based on their isoelectric pH, and then here they get separated based on their molecular weight, okay. This is how it will look, okay. Now look at it, so complicated. A typical picture, we have proteins, sometimes the problem is this could be several proteins all combined together, although we may think it is only single protein. So it may be very difficult to say this is single protein, and most abundant protein may come out as dark spots, others may come out very light spot. So it is quite complicated. If I want to now take out one of these and then run MALDI to find out the molecular weight and look at some sequence. So we have to be sure that this is only single protein and not a mixture. So theoretically these spots could be mixtures of protein, but this is how proteomics is carried out in two dimensional page, where we separate out in one dimension based on its isoelectric pH, and in the second dimension you separate out based on its molecular weight. So the proteins would have lost their activity here, especially when you run through the SDS page, but this is very useful technique if I want to look at the individual protein and characterize it in more detail actually. Okay, there are many softwares that are available by which can help you. It can compare with existing database and then say possibly this is the protein. So there are many softwares, like one of them is called Delta 2D. So it can use to quantify protein spots. That means by looking at this spot and by looking at the enhanced image, we can say protein concentration could be of this order. Match images, we can match it with the other images in the database and say this protein could be this much. compare corresponding spots and intensities of related gels. For example, I am having the protein from a tissue sample before treatment and after treatment. I can compare those two 2D gels, and then I say some proteins have disappeared. So, I can say maybe enhancement or suppression because of treatment. Prepare gel data reports, remove background patterns. So, for example, if there are proteins from one set and another set constant, we could remove them and just see those proteins which have appeared suddenly, okay, or those proteins which have disappeared suddenly after a treatment for a particular disease, and then go into databases and try to identify what that could be, or take out these spots. Okay, nowadays there are something called spot pickers, which can using an electronic system pick out a specific spot, and without a human touch directly inject it into a MALDI using a solvent, and then identify the molecular weight and the sequence of those proteins. So, such advanced studies could be done with the 2D gel, which I showed you actually. So, today we looked at a very important set of techniques which are normally used in molecular biology lab or biochemistry lab, trying to identify proteins based on the charge, based on their molecular weight and the movement of their diffusion coefficient and so on actually. So, what are the advantages, disadvantages of 2D page? Hundreds to thousands of polypeptides can be analyzed in single run, that is an advantage, right. Proteins can be separated in pure form from the resultant spots, because as I said, you can just pick up and then do whatever studies you want. The spots can be quantified, we can say the quantity in concentration, it can be analyzed using mass spec. Polypeptides can also be probed with antibodies. Yes, we can add antibodies and look at post translational modifications. We can look at differential expressions of protein between cell types. So, I can compare two cells and then say some spots have appeared, some spots have disappeared and so on. Disadvantages, large amount of sample handling, as I showed you there could be thousands of spots, okay. Limited reproducibility. If I take another sample from the same spot and run a 2D, I may get a totally different pattern, okay, and a smaller dynamic range than some other separation methods. So, not automated for high throughput analysis, it is very difficult for high throughput, and certain proteins are difficult for 2D page to separate, which are low in abundance. If the quantity is very low, it is difficult. Acidic, basic, hydrophobic, or very large or very small, we might not be able to identify in the 2D electrophoresis actually. So, these are the advantages and disadvantages of 2D electrophoresis actually, okay. So, thank you very much for this lecture.